said that we really need to engage the patients in our future discovery efforts and trials. We needed to engage our audience in this panel discussion. So I hope that you all are thinking about a lot of what Denny had to say here, because to me, he's opened up a whole lot of pathways of how we can go after proactively wellness and prevention. So to that end, I'd like, if I could, to ask Grace first, and then Peter, to introduce the areas of your focus so that we can get a little perspective on where you fit into the prevention / detection / treatment side of this discussion.
Sure, absolutely. You want me to stand up or sit? Either way? No, I'll just sit, that's fine.
No singing, though.
Don't ask me twice.
How about dancing?
After the cocktail hour. So thank you. Actually, that was a great setup for talking a little bit about my two companies, because they both deal with this aspect of chronic disease and the huge burden of care to the population. They're both progressive chronic diseases where a lot of the cost is driven by acute events. And so there's a huge opportunity, and the acute events also contribute greatly to the progression of disease, both in cardiovascular and pulmonary. And there's a lot of interventions that the patients themselves could give in terms of information that also guides their treatment and guides the progression of their disease. So both of these, they're very different companies. One's a diagnostics and one is therapeutics, but they have very common themes. So I'll start with Nkarta. Nkarta is focused on new delivery route to the heart for acute diseases, and that's by inhalation. So clearly, as you all know, basic anatomy, the heart's right next to the lung. Anything that is taken in by inhalation will go the pulmonary vein to the heart. And in the case of atrial fibrillation, which is a very common antiarrhythmic condition that actually affects over 30 million people worldwide, and in the US alone impacts $26 billion or more of direct and indirect costs to the healthcare system. 2 million of the folks in the US suffer from what's called the early stage of that, which is paroxysmal atrial fibrillation, which is a condition where your heart has an abnormal rhythm and can self-resolve within 7 days. But what usually happens, and many of you may have either experienced AFib or know someone who has, what usually happens is you feel terrible, you get palpitations, you feel weak and dizzy, and you just want to go to the hospital and have it taken care of. You want to go to the ER, you want to feel better. The problem is there are no good treatments for this right now. You showed some effectiveness data in terms of AFib drugs, and the oral drugs that are out there, antiarrhythmics, are maybe 50, 60% effective. And you have to take fairly large doses, and there are a lot of safety issues. So what if, and the other issue is the longer you're in that condition of arrhythmia, the harder it is to convert back to normal. So you have all these confounding effects. So you can go to the hospital, they might give you a large oral dose. Some people are taking these chronically, but you kind of become sort of immune to them over time. They don't really prevent the impact of the events. And then subsequently you end up, some percentage of the population ends up going electrical cardioversion or infusion IV drugs, which then are effective, but at that point you're in the hospital, you've incurred a lot of costs, you've been in discomfort and progressing for hours or sometimes days. So our idea was if you could take an inhaled formulation of one of these antiarrhythmic drugs, which actually works very well as an IV, but if you could take it and kind of carry around your inhaler, then you could control, the patient would have a lot of control. They'd be able to self-convert in a matter of 2 or 3 minutes. Now there's a dog model of arrhythmia, which is actually the gold standard by which all the antiarrhythmic drugs have been developed. And we showed in our model by delivery through the lung, you can convert in about a minute or 2 with a lot lower dose. So we are currently in phase 1 in Australia and in healthy patients. And we're very excited about this. And the other aspect that ties into this whole conversation about wearables and digital and personalized medicine is there are a lot more tools now whereby you can confirm that you are having an event. I have one on my phone. I can show anybody over the cocktail hour. They're made by other companies. There are patches. There's the Zeo patch. There are implantable devices. Medtronic has some. So that there's a lot of ways to track whether you are having an event or not and confirm it. The same goes for MI, by the way, which is our second product that we're developing, and I won't go into it right now. But the idea is you have this closed loop of being able to confirm that you're having an event, and ultimately even asymptomatic, which are the vast majority of events and are still causing damage to the heart and stroke risk, by the way. Eventually, you should be able to, if you know you're prone to this, you should be able to check every morning to see if you're having symptoms or if you're asymptomatic or even if you're starting to feel bad, and then take your inhaler and convert, and then check again and send all that information to your physician or to your healthcare system. And over time, ideally, we know that AFib begets more AFib, so if you can prevent these events and nip them in the bud, then you should theoretically be able to delay progression, reduce stroke risk, which is 15 to 20% of strokes are caused by AFib. So there is a lot of repercussions in terms of health and wellness and to the healthcare system. Now, the other company, Proterix, is looking at diagnosis and management of chronic obstructive pulmonary disorder and a whole a pipeline of other chronic pulmonary diseases. And the problem there, it's a very complex disease, $50 billion impact to the healthcare system, 14 million people in the US alone diagnosed with another 14 million undiagnosed. And it's a devastating disease. It's progressive. It accelerates every time you have what's called an exacerbation, and you're usually hospitalized. What if, and there's a lot of things going on in the body, inflammation pathways, fibrotic pathways, and so forth. It really lends itself very well to be better understood and managed by looking at a broad set of biomarkers that can tell you what's going on across comorbidities, populations, and different treatment paradigms and so forth. And so the idea of the company is, I think many of you heard Myriad speak this morning with regards to what the Crescendo test, that was a biomarker panel for rheumatoid arthritis that has many parallels to COPD, chronic progressive disease with flares, and that has a lot of things going on at the same time and are difficult to manage. Not a lot of good treatment, not a lot of good diagnosis options, spirometry and other things, so you know what's going on. But if you had a way to look at a disease activity index and know much more precisely whether you're nearing a flare or an exacerbation, the idea is across population you could better manage the disease. And so we have enthusiastic support from a lot of the KOLs globally in pulmonology about this idea, and we're collecting prospective samples. We're in feasibility, nearing validation, and hope to launch the first test in 2018. And we have a pipeline for cystic fibrosis and idiopathic pulmonary fibrosis. We also have a pipeline for neurology. We recently were granted a Michael J. Fox Foundation grant to look at early detection of alpha-synuclein. So we're working on, we're collaborating with Adriano Aguzzi at the University of Zurich for that. So both companies, and also in COPD, there are wearables, respiratory monitoring, patient diary tools that if you can integrate all that information, as you illustrated, we believe that with the biomarker information will be much more powerful and actionable. So there's both very big diseases, a lot of cost, a lot of opportunity to improve outcomes and save costs.
All right, so let me say a few words along when Kevin introduced the work I do, he mentioned the changing paradigm of testing, for instance, for cardiac troponin. In the lab I created and I direct at Brigham and Women's Hospital, we are performing a lot of testing of various cardiovascular biomarkers, one of them being cardiac troponin. And over the last 25, 30 years, the assay changed dramatically. We are really not involved in discovering new markers. We filed recently a patent application for an interesting marker of left ventricular remodeling, adverse remodeling of left ventricle. But most of the time, we try to understand how the changing sensitivity, performance of the assay, how it affects interpretation of the biomarker test results. And again, over 20, 25 years of cardiac troponin, this assay changed dramatically from being able to diagnose large MIs to small MIs with the current contemporary or medium sensitivity assays, with the upcoming high sensitivity assays, which not only scratch the surface of the so-called normal range or go beyond the diagnostic, below the diagnostic cutoff of those assays and can measure troponin in apparently healthy individuals. The use of the assays may change, the interpretation of the test results changes. current assays, the ultra-sensitive assays, such as the one developed by Quanterix or another one developed by Singulex, are so sensitive that you can reliably measure troponin concentrations in all healthy subjects, and that gives you a completely different idea of what this assay can tell you. So from trying to establish algorithms how to rule in and rule out MI, we are now looking at patients with stable ischemic heart disease, going deep into the so-called normal range and understanding that there is a marked gradient of risk for future adverse events, even in patients who are well below the diagnostic cutoffs of myocardial infarction. So it's becoming a very personalized, a very precise and very personalized way how to stratify patients according to the risk of future adverse outcomes. And again, for me, for instance, as much as for Kevin, my dad died of an MI at a relatively young age, and so it's kind of not only personalized but personal. And whenever I get hold of a high sensitivity, ultra sensitive assay, I can't resist to test my troponin, and I can tell you that with assays that have cutoffs at 26, I would hate to be above 10, because we know that once you go in the reference range or in this so-called normal range, that patients or subjects are at fairly high risk, even in the high single digits. So again, it's a very dramatically changing test with changing assay performance, and we are learning how to use it and understanding better as an integrator of overall cardiovascular risk, not just as a predictor of acute myocardial infarction.
Let me start. Debbie, you said something that absolutely was fascinating. You talked about taking 4 years to get an IRB and getting 135 patients recruited in a number of days. One has to say that that's really quite a striking contrast. What lessons are there, or how do you see the future in which IRBs don't take 4 years to get?
Yeah, I didn't say it took 4 years to get an IRB. It took 14 years for drug development, and we shortened that to 4 years, okay? But IRBs can be confounding. They are individual institutions, and they're small numbers. Those are the things we all face living in an academic environment, which is why, wearing my Pfizer hat, you're often going to a CRO in Germany or Australia or some other place so that you can do scale quicker and more efficiently. I think the lessons that we're going to learn going forward is that people, I don't even use the word patients anymore, people want to be participatory. Now, to start with, these are what I call elite performers. I don't mean physiologically elite, I mean participatory elite. But I don't worry about that because we still can learn a lot. We're developing algorithms from this diabetes subgroup, which is now totaling almost 60,000 people. And we have 65,000 comparable individuals in our own healthcare system, which is a combination of the Brigham and the Mass General Hospital. And we can begin to develop algorithms in an iterative way that allow us to stratify much more quickly. So that if I can send out an app and says, I want the following information, and we can extend that app. We're building clinical trial centers, both not only obviously in our institutions, but in partnerships organization. As I mentioned to many here, I'm a consultant to Verily, which is Google Life Science, which is literally across the street and 12 floors up from us here. We built a clinical trial center there, so people, volunteers, can actually come in and survey both the hardware as potentially getting samples for blood. This is the future. And what we need to figure out is how to develop those partnerships, which need to be public-private. As you know, there's a precision medicine initiative that was started by Francis Collins at NIH. It has the same kinds of concepts involved. So I think the lessons are that we need to get back we need to get rid of the non-scalable approaches that we have traditionally used in this venue for the last 25, 30 years and begin to build scalable ones. There are pitfalls and there'll be some hurdles to overcome, but I think we can get there, and I think we can get there sooner than most people think.
So, John.
Yeah, just related to that, have you seen any issues with getting consent from patients and data privacy issues, or do you see those as future issues?
No, this, again, maybe I didn't have time to go through this. With one click on the app, you've consented. Now, you can say, aha, boy, that sounds tricky. So we also ask a question at the end of, let's say, a 3-month period. We did it at 3 and 6 months, but it's 6-month period. Because one of the agreements we had with Apple is that they would never see the data, and that at the end of 1 year, we would be the only ones to see the data over a year. At the end of 1 year, any legitimate scientific enterprise could see the data. So it would be public within people who would be legitimately public. So we asked individuals, would you be willing now to share your data that you've shared with us? You've already agreed to share it with us, with the scientific group across the country or across the world. Now, if I had asked that question to my patients 5 years ago, probably 5% would have said, yeah, okay. The rest would have said, oh, no, 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 we don't want our data shared. 87% of the people in this study said, yes, we want our data shared. And part of the reason, again, is this behavioral aspect. On this app, you could compare yourself to everybody in Boston. You could compare yourself to everybody in Bangladesh if you chose to. So there were aspects of this kind of nuanced interrogation that the current society is used to, okay? How do I look at my internet? How do I do that? And they're very anxious to let people evaluate and annotate that information. Now, it's all done anonymously, so obviously that's part of it. And remember, this is clinical trials 101. I mean, we're simply collecting data. We have a continuous glucose monitor that is being developed with Verily. That would be the next step. And then the question is, how valid and legitimate would people want that data circulated? So we'll see. But I'm very encouraged by the first trials.
I have to say I'm very envious because at the Brigham and Women's Hospital, when we do a clinical trial, the consent forms are extremely complicated. And if we look for patients with chest pain, you can imagine that reciting 12-page consent form to a patient who is with serious chest pain essentially eliminates the patients whom we really would like to enroll, and we end up enrolling frequently patients who have heartburn and other conditions which we really didn't intend to study. So that really makes things extremely complicated.
Yeah, Peter's too young to know, but his boss, Gene Brunwald, and I helped write all those clinical trial. And as you know, anyone who's done a clinical trial or anybody from industry here know the iterative hurdles that one has to go through. And I don't care what kind of patient you are, when you get an 8 or 10-page document stuck in your hand, and remember, usually these are sick patients, these are not healthy patients, you don't want to get past page 1. And there's been this epiphany on the part of many investigators for decades about why can't we simplify this? We even are trying to get the idea that if you walk through a hospital like the Brigham or the Mass General, you simply sign a consent form. Now, it's not widely known that HIPAA actually allows you in an anonymous way to use medical data. That's why we can do studies on electronic medical records. We can do studies on discarded, piece of your colon or piece of your liver that gets discarded during routine clinical care, discarded blood. But that's all going to change. NIH is now going to insist that every one of those aspects get consented. So we could bring to a halt clinical research unless we actually figure out a more convenient and effective way to do this.
For those discarded ones, do you get the clinical data as well, at least as of now? Like all of the clinical data? I didn't think that was possible.
Up until now, you could take discarded samples anonymously.
Yes, sure.
So I could say, I'm a kidney specialist. I want everybody with a creatinine, I want a blood from everybody with a creatinine of 2. I don't know who those people are, I simply know what the creatinines are.
But can you get the de-identified, the rest of the information? That's important to know how that
Yes,
correlates.
yes. So then I can go,
Yeah.
we could go into what we call research patient data registry and get that information as well. Now, if I want to go and do experiments on those individuals, I have to consent them.
Yeah.
So one question I had for the group is just moving down the area of stratification. I'm curious if you look out and you're thinking about stratification either from some level of disease stratification and or treatment stratification and or prevention stratification, what kind of markers do you think are going to yield the most promise in the future? Like what areas of biomarker assessment do you think has the greatest potential? And I suspect the answer will vary. Like for instance, for your AFib arena, you might have a different set completely, but relative to proteins versus DNA versus RNA versus the microbiome, I'm just curious, like where do you think ultimately we're going to be able to really make a dent in the stratification?
Well, at the end of the day, phenotype is defined by protein expression, not what your genome is at any given point in time. But that's still a piece of the pie. It's a very big piece of the pie. And if you only could do that, and you guys are, the poster child for doing it well and doing it at levels that are meaningful, as you and I have talked about, below what we normally measure, that would be an enormous asset. But there are other assays that are going to become important. For example, as I said, we're finding a significant portion of the important metabolites of the microbiome are short-chain fatty acids. Now, not only do they engage toll-like receptors, but they engage orphan olfactory receptors, which exist, by the way, throughout the body. And the studies I weren't able to show you because they're not published, but the main reason we're able to now detect and quantitate type 1 diabetes is that these receptors engage and regulate Tregs. So for the first time, we actually have a pathway now where we can understand how a diversity of the microbiome, a set of metabolites, and a pathway engagement can lead to an autoimmune disease. My guess is this is going to be universal. We know, for example, some of the more recent advances in cardiac development is understanding choline metabolism that goes through the colon. This has been done at Cleveland Clinic, very elegant work. Much of that comes from red meat, so yet we have another annotation from things we kind of knew were bad, but bad because we were thinking only of saturated fats. We now know there are other components that when metabolized by the microbiome will lead to triethylene, choline, et cetera, that can stimulate inflammatory pathways. Those are the missing links that need to be measured. So we need to start with proteins. I'd say that gets you 75% of the way, and then the other 25% are going to be other small molecules.
I'm glad you brought up the microbiome because that's an area that I've always been a fanatic of. In fact, I got sort of involved with one of the early microbiome companies, which was using Affymetrix technology that was Phylotech, now Second Genome. And I think there are going to be enormous implications. It's kind of like when we looked at the dark matter of the genome at Affymetrix when I was there right around the time of the 2000. There's a lot going on we didn't know about because we weren't even looking for it. So the same is going to happen with metabolites and with microbiome. So there might be certain known biomarkers that we know to go after. There's going to be others that if we're not looking mechanistically at what's going and the interaction with the microbiome and the metabolites they're producing, we won't know what to look for. And so another example of that is epigenetics, right? So we have a company, one of my venture firm has a company in England that the co-founder of Celexa co-founded an epigenetics company that's looking at hydroxymethylation, which people weren't looking at before. And it has different implications in terms of what's getting switched on and off. And if you're not looking for that, you don't find it, yeah, ultimately it's going to be showing in the proteins, but you need to know what to look for. So you have other things to look for, whether it's microbiome, epigenetics, and other types of metabolites that will give you a clue as to what to go after and get a more complete picture. And that's, I think, a lot of advances are going to be made in the next few years in these areas.
I would actually posit that by 2030, 25% of all drugs will be targeting the microbiome. Remember, 8 million novel genes compared to 26,000.
And do we have sufficient tools in your mind today to get to the answers that you're looking for when you do some level of stratification detection of those metabolites and those?
Well, it depends what you mean sufficient tools. This field is in its infancy. As Grace said, even if 10% of what we currently now assume the microbiome is doing turns out to be true, it's going to be an enormously new organ system. Having said that, it's biochemistry. Measuring what the microbiome makes should be no different than measuring what the liver makes or the kidney makes, et cetera. Having said that, I'm talking in terms of the toolkits. In terms of the metabolites, they can be dramatically different. You got 8 million novel genes. Presumably, you're going to have some subset of that are going to be novel proteins or small molecules, et cetera. So I think that precision medicine, as you are trying to pursue it, needs to figure out how we're going to annotate something as massive and as diverse as the microbiome. And clearly, the way to do it is to get some clues. Like I said, the immune system obviously is important. And we start with that, but there's going to be a lot of other activities as well.
Well, big data will play a huge role in machine learning because you're dealing with so many interdependent factors, again, adding that level of complexity of a whole new system. And the other piece, everybody talks about targeting the microbiome with small molecules or maybe biologics, but what about the microbiome manipulation itself? If you had more standardized methods, GMP manufacturing, of measuring, reestablishing certain populations like people now do with C. diff, and fecal transplants, but in a much more structured manner, and be able to target to different areas of the body, repopulate, replenish, prebiotics. There's a lot to be done in that area as well, because with small molecules, you're going to have the same problem of off-target impact, right? So
yeah,
it's going to be hard to do. But
so for full disclosure, I sit on the board of Ceres, which
Yes.
is a microbiome company about 2 blocks from here, who is doing just that. The first trial with C. difficile was actually not a huge success. And part of the problem there is that I think it will be, and I think we will be able to, as my grandchildren laugh at, basically make poop in a pill that will eventually be able to treat not only C. difficile, but inflammatory bowel disease, et cetera. Having said that, we also now know that pathways are interrogated by the microbiome and its metabolites that clearly are part of the inflammation and injury pathway. Yeah. So I think we have to do both.
Oh, absolutely. Yeah.
Just one comment. When I became responsible for chemistry at the Brigham, my background was a lot of sequencing and cloning of genes and so on, and I thought that pharmacogenomics will be the thing that will take off within a few years, and I was ready to do all the pharmacogenomics and so on. And again, it's the phenotype which is so important to determine, because even complexity of the CYP450 subunits and their variance is such that an interaction of more than a few drugs is so complex that really it is the phenotype that we need to measure. I am still at this point, it hasn't taken off, and we are doing very, very, very limited pharmacogenomic testing after all those years, after 1.5 decades where we had all the tools to do it. But again, the interpretation is so complicated, and that it's the phenotype we really need to look at.
Hi. Thanks. Very good discussion. Greg Mayer, Biocore Strategies. Just a question. With all the advances that are going on with technological development in diagnosing disease and coming up with new ways to measure biomarkers, where do you see, as a panel, regulators and payers in this equation as these new advances go forward? One of the challenges, of course, with bringing new medical devices and technologies to market is initially reimbursement. I mean, initially getting them approved, but then also getting them paid for.
Yeah, this is, of course, the crucial question. Nothing breeds payment like success, of course, and I think it's a bit of a catch-22. We're going to have to show that pre-symptomatic assessment tools clearly save money. And if we can begin to drive that bus, I believe they will, whether it be in cardiovascular health or diabetic health. Cancer, again, is a good example where, at least at the level of genetic testing, but my guess is if we developed potent protein testing in cancer, pre-symptomatic testing, it would be a home run and it would be paid for. But we're not talking about science here, we're talking about politics. And I think at the end of the day, we're going to have to prove that whatever early diagnostic test we introduce in a pre-symptomatic manner will ultimately save money. And I think if we can do that and stratify patient populations so that they can also have effective medicine. I mean, the concept of effective medicine has been around since, I was an intern. We started the concept of, effective medicine and intellectually satisfying use of information. The problem is that 50% of what we do in medicine still is not substantiated by evidence. So the concept of evidence-based medicine is good for half of us, and the other half we guess. And that's a little hyperbole because we trust our physicians' experience to make these kinds of decisions. But the reality is that if we develop stratification and evidence-based medicine will become much more uniform around subpopulations, I truly believe that the payers, i.e. the government largely, will pay for these kinds of tests.
Yeah, it's going to boil down to both actionability as well as outcomes. So for a lot of these things, we're talking about changing outcomes and cost to the medical system. Right now, the incentives are skewed. So, I've been in all three sectors, genomics, diagnostics, and pharma. Guess who gets most of the money for improving the situation? The problem is, the opportunity actually is as we're moving to accountable care and more towards large employers and large payers and accountable care systems, there are opportunities, I think, to capture more of the value on the diagnostic and tool side of the value we're actually bringing by looking at populations and seeing if you're actually being able to decrease costs and sharing in that. And actually there's companies starting to plug into that model. In fact, we're looking at it for Proterix. If we're actually impacting that $50 billion a year cost for COPD, just reducing a fraction of that by reducing a fraction of COPD exacerbation events across a given population, there are opportunities to partner with accountable care organizations and other payers to basically say, look, we get no money if we don't save you any money. If we save you money, let's share in that savings. And it makes sense, and then you're more directly tied to those incentives. Right now, it's very skewed in terms of the way reimbursement happens, and so the investment in this space has been impacted by that. It takes a long time to prove the outcomes and tie it to actionability. And actionability is pretty important. I mean, there are a lot of cases where you might have the information, but if there's not something really actionable that you can do with it and actually impact outcomes, you're not going to get very far commercially. So there's a lot of exciting things going on, but everybody who is working in this field really needs to think carefully about the whole picture. You might have a marker or a series of markers that give you some information that you'd like to know, but if there's not a treatment or a drug or some sort of behavioral change or some sort of paradigm to change based on that information, it's not going to really get adopted or reimbursed.
I add a paradigm here that we also need to think more eclectically as to where the money is really coming from. Mm-hmm. With the concept of more and more bundled payments, accountable care, et cetera, there's a pool of money that can be exercised to invest in the proper test or the proper activity that will actually save the institution the money that they're getting from the government, the insurer, et cetera. Mm-hmm. I'll just give you a 30-second vignette. Everybody in this audience, and certainly everybody on this panel, knows that congestive heart failure is the epidemic that is going forward. When I was a young physician, people used to die of MIs. Now they progress to a chronic illness and they have congestive heart failure. Keeping patients with congestive heart failure out of the hospital is a multi-billion-dollar exercise. And indeed, if you get a readmission within 30 days, Medicare won't even pay for that. So we're under the gun already from the world's largest insurer, which is the US government, to keep these patients out of the hospital. So we invented a voice device. The larynx is a single epithelium on a muscle. It beats 900 beats per second, the fastest moving organ anywhere in the body. And a little bit of interstitial edema dramatically changes the harmonics. So we developed a voice device that you could use with an iPhone. You know, the brown cow jumps over the fence, et cetera, where we could predict congestive heart failure 6 to 10 days before weight gain. And using that, we can now intervene, give them a diuretic, whatever. The hospital will pay for that because they're losing $2 to $3 million a year for that kind of patient if we allow them to gain 3 pounds, which means 24 hours later they're often in the emergency room and we're treating them for pulmonary edema, which often means an intensive care unit stay, et cetera. So there will be a diversity of opportunity among the payers that will become more eclectic than what we're traditionally used to seeing.
I was about to mention heart failure as well from the biomarker point of view. Our understanding of natriuretic peptide is changing. While everybody feels that they are important tools for, or that natriuretic peptide guided heart failure therapy is the way to go, there are many clinical trials, but very few that were conclusive and demonstrated that that's really the way to treat heart failure. Definitely natriuretic peptides or soluble ST2 are great markers to predict the 30-day likelihood of 30-day readmission. So again, it's just one marker. We spent 15 to 20 years looking at natriuretic peptides, and there are certain things which we now understand better, but make things so much more complicated. Glycosylation, degradation, and so on of natriuretic peptides is a whole new area where we actually are looking into developing even better biomarkers than natriuretic peptide, BNP, or N-terminal BNP, pro-BNP. So again, it's an extremely complex field, but even for such a simple thing like heart failure with reduced ejection fraction, it's still not definitely proven that natriuretic peptide guided therapy is the way to go.
So I'm going to maybe close with one last question, and I'm not sure exactly how to ask this, but it seems to me that when we look at the socioeconomics and we look at the demographics of obesity that's going on in the United States, as well as overweight being as much as 2/3 of the country, that's fueling inflammation and fueling a lot of disease, it's pretty easy to see a paradigm where we're driving a lot of these disease pathologies earlier and earlier in life with this kind of frantic kind of trend that's occurring. And I just wonder, from where people like the three of you sit, it's not probably as obvious to you why we should, from what your expertise is, that the prevention side of this has got such an incredible opportunity, because that's not really necessarily what you're geared towards. You're probably geared towards early detection technology and assessment and stratification and maybe drug discovery. But I'm just curious, like, what your views are on this whole concept of being much more scientific in preventative sciences and trying to stratify and really try to train and shift the obesity crisis, like Japan and other countries have done, and significantly shifted their healthcare expenditure as a result of it. And I was just curious if you had any views on that.
Well, I think as a physician, that ought to be our ultimate goal. I mean, when I was in medical school here, 400 years ago, preventative medicine was a 5-minute lecture in medical school. Stop smoking and wear seatbelts, and that was pretty much the extent of it. We haven't gotten too much further since then. Obviously, diet has become the hardy perennial now in thinking about this. But the reality is pretty straightforward. We as human organisms have a makeup biochemically that's 100,000 years old. And that's how we evolved. And our biochemistry was geared to surviving when, you know, hunting a saber-toothed tiger wasn't so easy, and maybe you got protein every 3 days, and there were no grains. There was very little else that you could count on. So socio-economic development far exceeded biological development, and here we are in the 21st century with McDonald's. As I pointed out with type 2 diabetes, I mean, do you realize that whether I go to my clinic today or Peter goes to his clinic, almost every patient that walks in with hypertension, diabetes, obesity, a simple lifestyle change will cure, literally cure, 75% of those diseases. I mean, you can't say that about anything else, okay? So of course medicine ought to have that as a primary focus. The problem is that it's 80% behavior, and that's why we focused on developing behavioral apps. We are creatures of behavior, and the question is not only how you measure behavior, but how you modify behavior. But there's no secret. If I had every one of those patients lose 3 to 5% of their body weight, their blood pressure would fall, their blood sugar would fall, a whole variety of other risk factors from obesity, for women, breast cancer, for men, colon, prostate cancer, etc. All those things would dramatically diminish. So there's no secret. The question is how to achieve it, and how to achieve it in a socioeconomic environment where there's a lot of Big Macs sold today, you know. Yeah.
Yeah, I've been thinking a lot about this. This is an area I'm really passionate about, and I think there are a number of issues. There's behavioral issues, there's policy issues, and there's financial incentives, which tie to behavioral issues, both at the policy level and at the individual level. And I think that there, you know, Kevin, you alluded to it beautifully this morning in terms of this whole, the subsidies for sugar and grains, which then have all these knock-on effects, both in terms of the packaged good industry putting sugar in everything, even in savory items, and all the havoc that wreaks. In terms of what we feed cattle, which then screws up the omega balance, and then also the environment and everything else. So there are so many follow-on effects that start policy-driven in terms of how things are subsidized and what the economic incentives are. But there's also hope, I think. There are companies that are looking at behavioral modifications. Omada Health, for example, is a great example. They actually get reimbursement for their preventive care partnership with companies, and where they actually help and interact with the employees in terms of diet and monitoring and exercise, and there's financial incentives and other kinds of incentives involved, and they're starting to see results. And I back to the microbiome because I can't stop. I do think that there are some aspects that are lifestyle, other aspects are genetic and environmental, like microbiome. So all the studies, and you know much more about this than I do, but the studies I've seen where they look at, they implant the feces from skinny mice into obese mice and they get skinny. There are microbiomic profiles that are correlated with higher metabolism and different types of things like that. So I do think that there may be some stratification opportunities involved. It could be very consumer-driven by people motivated to improve their health, where you might be looking at microbiome seeding and you know, prebiotics or whatever it might be. I don't know what the answers are. But I do think that a combination of policy changes, financial incentives, and behavioral motivations, and you know, for payers, there's a huge incentive, right? If they can tie it back to a population that they control, and there's ways to work with companies like Omada and others who are doing these types of things. And then also we have to do at the policy level, at the government level, in terms of where our subsidies are going.
Yeah, there is not that much I can add. I can only agree it's education, education, education. Lifestyle change. Coming from Europe, maybe a little bit less of political correctness, kind of calling things what they are. Obesity and overweight is obesity, overweight. It's not new standard of beauty as we can see it frequently in journals and other media outlets. So yeah, that's again, lifestyle change is absolutely crucial. It's nothing else we can do. I can measure troponin as much as I want, but if that patient doesn't lose a few pounds, it won't help.
Well, I would love to end it on that high note of prevention, because I think those sciences are going to continue to evolve. And I cannot thank Denny and Peter for sharing their views of the heart and cardiovascular, and thank you very much for your time.